Hello all, welcome back to the BioRocks channel. In this video today, I'm going to teach you regarding the chemiosmosis theory or chemiosmotic hypothesis. Okay, we are going to learn about chemiosmotic or osmosis hypothesis. Okay, this was being proposed by Dr. Peter Michel. Dr. Peter Michel was a person. He has proposed this theory in the year of 1961, but this was not been accepted by the science communities. Okay, but later in the year of after having a patience long wait of 1976, I think it's all around like 15 years of patience. He has received the Nobel Prize in the year of this particular 1976. What actually this speaks is about, for instance, what he was proposing was ATP molecule, which is an energy-rich molecule, gets generated when there is a flow of protons from higher concentration to lower concentration. Okay, lower concentration. This results in formation of ATP molecule by an enzyme which is being called as ATPase, or we are going to call this as ATP synthase. This is as simple as such. Just remember again, the movement of ions across the concentration gradient from a higher concentration to the lower concentration. Which molecules or ions are moving? The ions are the H plus is nothing but the protons. Okay, the protons are jumping from a higher concentration to the lower concentration through a semi-permeable membrane. Okay, this is going to result in formation of ATP by an enzyme, okay, utilizing an enzyme called as ATP synthase or an ATPase, okay. So this is being called as actually chemiosmosis hypothesis, but many of the science communities haven't actually what you call is accepted this theory, because according to them it was not ATP, but according to them it was some intermediate molecule which was actually what you call is trapping up the energy during the jumping of the protons from a through down a concentration gradient. It was an intermediate molecule, but they haven't accepted that this movement of the protons from a higher concentration to lower concentration actually has resulted in an ATP molecule by this enzyme. But according to them, it was the energy was being present in an intermediate molecule. Okay, but in the year of 1976, this was being proved right, and he was being awarded Nobel Prize in the chemistry department for this particular chemiosmosis hypothesis. So I hope children you have understood what is chemiosmosis is nothing but the movement of ions. Let me write this for you. The movement of ions down a down a electrochemical gradient. Down a electrochemical gradient. Okay, what is electrochemical gradient, children? Electro is nothing but because they have charges. Okay, the chemical is nothing but you know that the concentration of these are higher here and lower here. So when you speak about the concentration differences and the number of ion differences across the semi-permeable membrane, we should call that as a electrochemical gradient. For instance, imagine if there are many protons this side, the number of the negative charges are less, but the dominating are the positive ones. This side, the positives are less, whereas this side it is going to be net negative charges are more. So now what happens is, to maintain the equilibrium, the protons from the higher concentration will be moving towards the lower concentration, and this will be utilized to produce or synthesize the ATP molecule by an enzyme which is being called as ATPase or ATP synthase, children. Okay, this happens both. The chemiosmosis is going to happen where, it is going to happen both in the mitochondria okay, as well as the chemiosmosis, because during mitochondria we are generating, that is cellular respiration process, we are generating 38 ATP molecules from glucose. So production of ATP is nothing but what? Production of the ATP is called as phosphorylation process. So a production of ATP means it is just because of the movement of the protons down the concentration gradient is going to generate ATP because, according to the chemiosmosis. So when we speak about cellular respiration, when we speak about the cellular respiration, we speak about what? Chemiosmosis process, okay? And when we speak about the chloroplast, that is in the photosynthesis process, so even inside the chloroplast also, you know that during your light reactions, I told you that there are production of ATP molecules as well as energy currencies which are being called as NADPH2s. This we have seen in the what you call is photosynthesis process. So in the light reactions, you can see the cyclic and the non-cyclic chains which are being seen. So during that also there is ATP production. So when we speak about the ATP production, then we have to speak about which process? These two. So here also ATPs are being produced. So we have to see that this procedure is going to be something which is related with the phosphorylation process. So children, just once again in simple terms, I'm telling you that when you speak about the phosphorylation process, this process speaks about the production of ATP. This speaks about the production of ATP. When you speak about the production of ATP, then we have to think that there is compulsorily chemiosmosis process which is driving this procedure. Okay, who is driving ATP production? This is chemiosmosis. So phosphorylation is happening in which processes here? It is happening in the cellular respiration process as well as it is happening in the photosynthesis process. What? Phosphorylation. So even cellular respiration is going to show what? Chemiosmosis. Even photosynthesis is also showing what? Chemiosmosis process. For what purpose? In order to produce ATP. In order to produce ATP, we are running the chemiosmosis both in the cellular respiration, both in the photosynthesis. What is chemiosmosis involving? It is moving the protons from a down a gradient of what you call is from higher concentration to lower concentration, where this energy is utilized by the ATPase to generate the ATP molecule by combining the ADP plus inorganic phosphate molecule. During this movement of ions from the higher to the lower concentration, the energy which is released is utilized by the ATPase to produce ATP, which is being called as what? Chemiosmosis process. I hope children now you have understood. So to make this procedure very easy for you, what I'll do is I'll explain you the electron transport chain. Then you will understand this procedure. See children, another thing, where is chemiosmosis happening? Osmosis is going to happen wherever there is electron transfer. Wherever there is electron transfer, or wherever you speak about the electron transport chain, wherever we are going to speak about the electron transport chain, there we can see that the chemiosmosis is going to happen. Okay, did you understand this? This is one of the simplest hints what I can give you here, children. Okay, have you got it? So remember that the electron transport chain is nothing but here. Where have you seen this electron transport chain? In the cellular respiration process, we have seen the electron transport chain, okay, after the Krebs cycle. And even remember that the electron transport chain even is seen in the non-cyclic photophosphorylation, right? Non-cyclic as well as the cyclic photophosphorylation processes. In the photosynthesis process, we can see that there is electron getting transferred from one carrier to the other carrier. That is nothing but the electron transport chain. So even in the cellular respiration process as well as the photosynthesis process, there is electron transport chain, and there we can see the chemiosmosis happening, which is going to result in the formation of ATP. I hope children I made it very clear, crystal clear for you that what is chemiosmosis, where it is getting involved, what is it going to produce. Okay, now let me take a fresh page and I'll explain you this particular chemiosmosis process, for which what I'll do is first of all I'll draw a mitochondria for you. Okay, this is going to be our mitochondria, where you can see that the mitochondria is going to have these inner membrane and an outer membrane, and they are going to have these folds which are being called as a cristae, because they are going to increase the surface area of the inner membrane, because this is very important for our electron transport chain. So children, what I'm explaining you is the electron transport chain, because I told you wherever there is electron transfer, what happens? Chemiosmosis happens. Just remember, children, that here what I'll do is we are going to have these what we call is different integral proteins. You know that your outer membrane. Let me first give these names for you. This is the outer membrane of your mitochondria. Okay, and this is going to be your inner membrane. This is going to be our inner membrane, and the folds are called as cristae. Okay, and now the space between the outer membrane and the inner membrane is being called as what? Intermembrane space. Okay, intermembrane space. So the space between these two, outer and the inner membrane, is called as what? Intermembrane space we are going to call. Just get an idea that your inner membrane, which is being present here, it has different complexes. Okay, protein complexes. We call them as the enzyme complex or electron complex. So let me draw those complex for you. Okay, see, now I'm drawing on the inner membrane. We have a special electron yeah, what we are going to call as, it is having an enzyme complex, and the name of this complex is number 1. Okay, secondly, it is going to have another complex on the inner membrane, and this complex is being called as complex 2. Okay, and they are going to have another complex in the inner membrane, and this complex is being called as complex number 3. And next, in the inner membrane, we are going to have the complex which is being called as complex number 4. These complexes are nothing but the electron complexes. Okay, these are the electron complex, or you can even call them as a enzyme complex, which are made up of proteins and some enzymes embedded inside. So what are present here are, there are some enzymes also which are being embedded here, children. Okay, so let me just give you that here is the enzyme. Okay, this is going to be the enzyme which is embedded, and this is going to be the enzyme which is being embedded over here. Okay, what are these complexes? These are the electron complexes or enzyme complexes of the electron transport chain. Where are they present? On the inner membrane, or in the inner membrane, what I can say is. So now what we are going to do is, let me take a suitable color. Yeah. Now there are some other electron carriers which are present here in the inner membrane, and these electron carriers are none other than coenzyme Q. So this is going to be called as a coenzyme Q. Okay, coenzyme Q. Coenzyme Q can also be otherwise called as ubiquinone. We are going to call this as ubiquinone. These are not the protein complexes. Okay, these are not protein complexes. As you know, coenzymes are going to be like a vitamin, what you call is compositions. Okay, and this is a movable one. These are movable. These are movable along the what you call is a membrane. What is a membrane made up of? Lipoproteinaceous, bilipid layer. So even your outer membrane is a bilipid layer, inner membrane is also a bilipid layer, and your inner membrane, which is a bilipid layer, is been having its own integral proteins. Okay, every bilipid layer is going to have what? Its own special integral proteins, and those protein complexes are nothing but your complex 1, complex 2, complex 3, complex 4. Okay, and we are even having the electron carriers like coenzyme Q in between the 1 and 2. Okay, and even we can see that there are some other ones which are being called as a cytochrome C. Cytochrome C, which is going to have what? Which is, sorry, this is going to be only present inside the inner membrane. Okay, do not consider the outer, only the inner membrane. And this is going to contain iron as a main central atom over here. Okay, this is very, very important for you all children. Did you understand? So your mitochondrial inner matrix is going to have integral protein complexes like 1, 2, 3, 4, what we call as electron complexes or enzyme complexes of the electron transport chain, And remember, there are electron carriers like coenzyme Q and cytochrome C also present here. What is the inner thing matrix here is being called as a stroma. And let me take a fresh page and give you the, okay, this is what I'm drawing about. This is our inner membrane, children. Give me a moment. This is going to be our inner membrane, okay? This is our inner membrane of mitochondria, the bilipid layer, the bilipid layer. And this is going to be your stroma side, okay? In the stroma, which cycle was running actually? Krebs cycle. And you know that glycolysis and Krebs cycle has resulted in what? The DH2 molecules as well as FADH2 molecules were produced, right? These were being produced during the Krebs cycle. This is your inner membrane. And this is going to be here, this is going to be the outer membrane which is present, okay? This is also going to be a bilipid layer. This is also a bilipid layer. So the space between the outer membrane and the inner membrane is being called as what? We are going to call as a intermembrane space. This is being called as a intermembrane space. Now, children, I told you that there are inside these inner membranes, there are some protein complexes, I told you, right? These enzyme complexes. So I'm drawing it. This is complex number 1. And we are going to have another one, which is going to be called as complex number 2. And I'm having another one, which is being called as a complex number 3. Okay. And I'm having in the inner membrane other complex, which is being called as complex number 4. And then I'm even having another enzyme which is going to be the what you call is ATP synthase, which is going to look like this, which is stalk with a stalk as well as a rotor which is being present, okay? A rotor which is being present. So now you see that these are the complexes, and I told you that these complexes, in order to have a proton pump, I'm drawing this structure here, okay? So remember, children, they were even possessing what? They were even possessing these electron carriers which transfer the electrons, okay? They were none other than the coenzyme Q also present, as well as there is another one which is being present inside the inner membrane, which is being called as cytochrome C. Now, children, what I need to speak here is about the, what we call is a Krebs cycle here. This Krebs cycle was continuously providing you not only Krebs cycle, but also we saw that the glycolysis produced 2 of them, and we have got few of the NADH2s, NADH2s produced over here, as well as 2 FADH2s also produced during the Krebs cycle. So now what happens is there the enzyme complexes. I haven't drawn the enzyme complex. Let me draw the enzyme complex for you. So not this color, let me take another slide, another color. Yeah, see here in these complex 1 and the complex 2, there are some enzymes associated here, okay, enzymes. Now, see children, these enzymes, what they have is they have a stronger affinity for these particular electrons, okay. So the complex 1 has higher affinity for the NADH2, okay. So now what happens is NADH2 is going to approach the complex 1. As this enzyme is having the highest affinity towards electron, it pulls the electrons from the NADH2. So now it has pulled the electron from the NADH2. So what happens is, since it has lost electrons, it is going to get oxidized, and it is going to even release the proton also. Okay, what has happened, children? In the stroma, as continuously there are NADH2s and FADH2s produced here, and we know that the complex 1 has an enzyme which has a stronger affinity for the NADH2. So the NADH2 got pulled over here, and its electron is been ripped off from, and it has got oxidized. And now what has happened? It has even released what? The proton, because every hydrogen atom is going to have what? It is going to have a proton and a electron, one electron and a one proton. So one hydrogen atom, if it is being pulled, one electron is being ripped off into the complex, and one proton is going to be released into the stroma. Since here two hydrogens are there, it is going to be two electrons which are being released, and it's going to be two protons which are being released into the stroma side. So now the stroma side, what is happening? Protons are added, and what are added into the complexes? Electrons are being added. See children, what happens is, now when you are going to see that it is going to take up the electron, the complex 1 is going to get energized, okay, remember. So now what happens is, if this is going to be the state of energy of your complex number 1, if this is going to be the complex number 1 energy state, now what happens, you see. So as it is going to accept the electron, let me change the color now. as it is going to accept the electron, now what happens is the energy level is going to raise up. But any molecule cannot stay in a higher super excited state for a longer time. So what it does is it is going to lose the electron and it comes down to what? The normal ground level. Okay, this is going to be the ground state. Now this is going to be the ground state. So now what has happened to the complex one is it accepted the, it took the electron from where? From the NADH. And what has happened is the electron, as it took up the electron, it went to a higher energy level, and later it loses its electron and it comes back to the ground state. During this procedure, what happens is, children, it is going to release energy. So this procedure, what it does is it is going to release energy. So this is doing what? This is releasing the energy. This process is going to release energy. Now what happens is, as this particular thing, which we are going to call as energy excitement, okay, electron aa gaya hai, so it has got excited. Now it has passed these electrons to whom? To the coenzyme Q. So the coenzyme Q electrons are being given. Now what is happening is, as it got excited and it lost the electrons, now the complex molecule is going to get changed. As it is going to lose electron, the molecule, the complex molecule is going to change. The molecule change is change in what? In the structure. So now what happens is the pores open up, and slowly what happens is the protons which are being present from the stroma side are going to be picked up. So if this is a proton, the proton will be picked up from this particular pore and it will be released into the intermembrane space. I hope you understood now. Okay, so this enzyme pulled up the NADH2 and it has ripped off the electron from the NADH, it has oxidized it, and it releases the protons towards the stroma side. And once it is going to get energized, what is happening? It comes down to the normal ground state by donating the electrons to one of the electron carrier. During this procedure, the change in the molecule structure changes, okay? And now what happens is the proton is going to be pumped out from this particular procedure. So if you remember, when I've taught you about the electron transport chain, I told you that when the electron jumps from one carrier to the other coenzyme, what happens? Energy is released. That energy is utilized for what? Utilized for the proton pump. So now that energy is utilized to pump the proton from the stroma side to the intermembrane space now. Got it? So now it went into the coenzyme Q. Now what about this particular FADH2? FADH2, the complex 2 is going to have a higher affinity towards the FADH2. So FADH2 does not approach the complex one. So what is happening is the FADH2, as the enzyme present in the complex 2 is having affinity towards this, what happens is it pulls away electrons from these particular, what we call as the FADH2, and then what it does is it is going to oxidize this particular FADH2 into what? FAD plus, and it is going to even release the protons towards your stroma side. Okay, so now what happens is the complex 2 gets energized. Okay, it gets energized, but I haven't drawn any pump or core for this. So it can't, what we call is pump up the protons, but what it does is it is going to donate its electrons to the coenzyme Q. Once again, children, the complex 2 is not having a pore to pump the proton out, okay? It just transfers the electrons to the electron transport material, which is coenzyme Q or a ubiquinone. And now what happens is these electron carriers, I told you that they are non-proteinaceous. They can move inside the membrane. Now what they'll do is they will just transfer these electrons to this particular complex number 3. Once the complex number 3 is going to get the energy, what happens? It is going to electron, it as it accepts the electron, it gets excited and it loses the electron. Its molecular size, that is a confrontation, is going to change. And now what happens is it is going to come down to the ground state by releasing energy. So the same thing happens here. What happens is this is also going to have this particular, what you call as a pore, from where what it does is, as it is going to get energized by receiving the electron here, now this is going to pick up the protons from the stroma side and it is going to pump the protons into the intermembrane space. So many of these will be going on getting released from the complexes number 1 and complexes number 3 here, and the number of the proton concentration concentration in the intermembrane space is going to just go on increasing. Okay, now what happens is this electron is being kicked up into the cytochrome C. Cytochrome C. This is also an electron carrier. And now what happens is the cytochrome C now is going to transfer these electrons into the complex number 4 now. Okay, and this complex number 4 is also going to contain what? The pump. Now, as it receives an electron, it gets energized and the molecule shape is going to change. And now this pore is going to pick up the protons from the stroma side, which are getting released from these two, what you call is oxidized coenzymes, and the protons are going to be pumped into these particular things. But now what happens is, where does this electron go now? This complex 4 is going to be the last complex present here, and now these electrons will be given to the last electron, what you call is a receiver or acceptor, which is going to be the oxygen. And now once this is going to accept the electrons as well as protons, okay, the oxygen not only receives the electrons from the complex number 4, but also it receives these what you call is the protons from the stroma, and it is going to get converted into the water molecules. Okay, water molecules. Now what happens is your protons which have been built up here, they are in high concentration here, and the protons here are in the low concentration here. So we know that according to the chemiosmotic pump, the protons have to move from a higher concentration electrochemical gradient to the low chemical electrochemical gradient. So now what happens, children, is nothing but this is going to see, I have to say that this is going to be your ATP synthase, ATP synthase, or we are going to call it as ATPase. Now, what happens is, as the protons are going to be moved from this particular ATP synthase, what happens is your ATP synthase is going to be rotating. It's going to rotate, and the kinetic energy here is going to be utilized. This energy, the energy which is rotating your ATPase enzyme is being utilized to add up an ADP with inorganic phosphate molecules to produce your ATP. This is going to be the procedure of your electron transport chain. I hope you all have understood, children. Just remember that as the complex 1 and complex 2 are going to have a stronger affinities for the what you call is NADH2 as well as FADH2s over here. What happens is these particular ones, they approach the complexes, they lose their electrons into the complexes while they lose their protons into the stroma. Okay, once their complexes receive the electrons, they get energized and the shape of the molecule changes and the protons are being pumped out. Okay, how are they being pumped? Because when the complex by receiving the electron comes to a higher energy level, it loses that electron to one of the what you call is electron carriers. This emits out energy. That energy is used to pump the proton from the complexes into the from the stroma side to the top here intermembrane space, and this is going to result in the accumulation of the protons in the intermembrane space, thereby increasing the concentration gradient. And from a higher level, now they enter through the ATP synthase, rotating the particular enzyme and creating energy. That energy is being used to bind the ADP with the inorganic phosphate molecule to produce this particular energy, which is being called as ATP. I hope you have understood, children. Let me come to this particular previous pages where I was telling you that these ones, now you see what has happened. Wherever there is electron transport chain, I told there is chemiosmosis, pumping of the protons from higher concentration to lower concentration through a semi-permeable membrane. That semi-permeable membrane is none other than your inner membrane of your mitochondria, okay, that resulted in the formation of your ATP, okay. Now what I'll do is, children, let me share my screen with you all for showing you few of the slides which will give you more information regarding this, okay, that will be helping you in memorizing during your examinations. Okay, the first thing that I would like to show you is going to be the electron transport chain that I have explained you now. So if you're going to see this, children, this is the outer membrane, okay, and this is going to be the inner membrane here. Coming on to the inner membrane, we are having complex number 1, the complex number 2, the complex number 3, and the 4, and then lastly we have ATP synthase, which is being present here. Remember, children, if you're going to see the NADH, please correct it. I have given you NADH2, that is NADH. Please do correct that particular thing, okay? So Na DH, which is a reduced coenzyme, this complex 1 has affinity for it, so it gets approached here, and the enzyme complexes present in the complex 1 are going to pick up the electron, and the electron is going to energize this molecule, and it will releases that electron to the one of the electron carriers called ubiquinone, and later the change in the molecule results in as well as energy which is released is used in pumping the protons from the stroma side to that of your intermembrane space. From coenzyme Q, the electrons, as well as you can see the FADH2 is that is complex 2 is having higher affinity towards this. It gets approached over here. Now what happens is the electrons are being pumped into the 2, but this does not actually pump up the protons, but it is going to pump the electrons to the coenzyme Q, and the electrons from the coenzyme Q are going to be passed to the complex number 3, where we can see that the energy is used for pumping the protons out from the stroma into the inner membrane, as that is intermembrane, and you can even see that the electrons will be passed to the electron carrier cytochrome C, which has iron as a main central atom. From there, the electrons are pumped into the complex number 4. Again, the protons are being pumped by using the energy, and electrons are at last being transferred into the oxygen. Not only the electrons from the complex, but also from the protons, even from the stroma, are picked up by them, and it results in the formation of the water. And the ATP synthase is going to pick up the protons from a higher concentration gradient, and as they pass on, this is being rotated, and this energy is being utilized in binding up the inorganic phosphate with the ATP, that is ADP, to produce your ATP molecule. This pumping of proteins from higher concentration to lower concentration, the chemiosmosis, which resulted in the formation of the ATP, but not any intermediate molecule, children. This is what I needed to show you from the electron transport chain. Now let me show you the same thing, but let me give you the names of the enzymes or the complexes. The complexes number 1 is going to have an enzyme called as NADH reductase, okay, and the complex 2 is going to have succinate dehydrogenase, which has higher affinity for the FADH2, okay, and the cytochrome, the complex number 3 is going to have an enzyme called as cytochrome reductase, and the complex 4 is going to have the enzyme called as cytochrome oxidases. Okay, these are not required, but just remember that the enzymes and electron carriers and the complexes are the ones which are actually taking up transferring the electrons, and during transferring the electrons, the energy is being utilized for pumping the protons, and these proton movement from a higher concentration to lower concentration through ATP synthase will be resulting in the formation of ATP. Just remember, children, that oxidative phosphorylation means what? Production of ATP by the ADP pi reactions and energy in presence of oxygen. So this oxidative phosphorylation in our cellular respiration has electron transport chain as one unit, as well as chemiosmosis as a second unit. This is very, very important to remember. Okay, where is ATP generated? ATP is generated inside the stroma. Now, coming on to the same thing, if you're going to look at the what we call is a that is a photosynthesis. So before I speak about photosynthesis, children, how the electrons are being transferred, you need to know what is photosystem. A photosystem is nothing but the main pigment, that is chlorophyll A, along with its accessory pigments like chlorophyll B, the carotenes, as well as the xanthophylls, all together. You see, all of them individually are also absorbing a particular wavelength of light, and even the chlorophyll A, and they are also, what they do is they're going to actually send it back and concentrate it to the chlorophyll A. Okay, so we together along with the main chlorophyll A pigment as well as axillary pigments togetherly have to be called as a photosystems, we are going to call. So now we'll see how the electrons are transferred in the photosynthesis, and how chemiosmosis is producing the ATP molecules in the photosynthesis process, children. Now you see, this is going to be a thylakoid membrane. Thylakoid membrane. As you know, light reactions are happening in the discs. There's a grana is nothing but the thylakoid membranes. We are having these photosystem 2 as well as a photosystem 1, which are having the axillary pigments as well as a chlorophyll pigment. They are fixing the light, okay? They are absorbing the light quanta, and the electron is going to get energized, okay? And that electron is going to be thrown into the several electron carriers which are present, like plastoquinone, cytochrome, plastocyanine. These are all the electron carriers which are being present. So once the electron is being kicked out, the excited electron is being taken up by these all electron carriers and it's been transferred. While electron is going to be transferred from one carrier to the other carrier, what happens? They are going to get energized. That the energy is going to be released. That energy which is released is utilized in what? Proton pump. Okay. Once again, I'm trying to repeat this. The quanta has energized your electron. It gets excited. It is being kicked out, and that electron is going to move to the several electron carriers. When it moves from one to the other, there is energy released. That energy is being used in proton pump. Okay. So now what is happening is the protons are pumped into where? Into this particular thylakoid lumen. Okay, thylakoid lumen. Secondly, you can even see photolysis happening. Photolysis also releasing the electrons. See, the electrons are being taken up into the photosystem too. And what is happening here? Protons are also being released, right? Because hydrogen has one electron, one proton. So as electrons are being released into the photosystem too, same way the protons are being released into where? Into your stroma side. So continuously there is built up of many proton molecules inside the thylakoid lumen just because of the photolysis process now. So now what is going to happen is, as a concentration of the protons are going to be more here, the protons are going to be moved through the ATP synthase. Now what happens is, children, the ATP synthase is going to spin and the energy is used to combine the ADP with the inorganic phosphate to produce what? ATP. Which side is that? Stroma of your chloroplast. This is your thylakoid lumen. So in the thylakoid lumen, there is a high concentration of the protons because of the continuous photolysis process. As well as the protons are being pumped from here during the electron transfer. So now the protons will be pumped from the thylakoid lumen to the stroma side and the ATP is produced here. But remember that not only ATP, the assimilatory power is also NADPH, if you remember. Okay. So now what is happening is, children, here, the thing here, what is happening is, when the photosystem 1 is going to get the electron from these electron carriers, as well as the light which is going to excite the electrons here, are passed into the ferridoxin, which is also an electron carrier. Okay. And this ferridoxin, when it is going to lose electron, it loses its electron to the NADP plus. Okay. The oxidized coenzyme. And now the oxidized coenzyme is going to get reduced to NADPH. So these are going to be the assimilatory power. So you can see that even in the photosynthesis in the thylakoid membrane also, during light reactions, there is electron transport happening, during which there is proton pumps. And these proton pumps are going to compulsorily build up a higher concentration on one side, which results in the chemiosmosis, during which the ATP synthase is going to release a ATP molecule. This is important. I hope, children, I made it clear for you all about the chemiosmotic pump. So if you have understood this particular video, I wish that you all, children, please hit a like button if you liked it, and please subscribe my channel and try to share this channel with many all. Okay. So just remember, children, the points I told you before I end up my video. Just remember that when there is a proton or an ion gradient or a charge difference gradient which has been built up, is going to result in the movement of ions across, that is called as electrochemical gradient. The electrochemical gradient is going to result in the production of the ATP molecules, especially during the electron transfers or electron transports, whether it might be the respiration or whether it might be the photosynthesis process. That is going to result in the production of ATP, which is nothing but the oxidative phosphorylation in the cellular respiration and photophosphorylation in the photosynthesis process. Okay, children, I hope you all have understood this particular procedure. If you like this video, please hit the like button and subscribe my channel, and please share this channel to many other children too. Okay, thank you all.